VinFuture Prize, a new global science and technology prize for humanity from Vietnam. One VinFuture grand prize of $3 million. 3 additional special VinFuture prizes valued at $500,000 each. VinFuture Prize honors science and technology work that creates or has a high potential to create meaningful change in the everyday lives of millions of people. Join us to make a change for a better future.
Good morning, good afternoon, and good evening, wherever you are joining us from. It's a pleasure to have you with us for the first InnovaTalk webinar of 2025. I'm Vi Dương, and it's my honor to be your moderator for today's sessions. And now, we'll set out to explore the transformative field of regenerative medicines, with the world confronting increasing healthcare challenge from aging populations to chronic illnesses and critical injuries. Innovations in regenerative medicines can open new doors to long-term healing and enhance well-being. Uncover the vast potentials of this groundbreaking field, the VinFuture Foundation is proudly co-hosting the InnovaTalk webinar on regenerative medicines. And prior to commencing today's webinar, I wish to extend our sincere appreciations to the European Molecular Biology Organizations, or EMBO, for the partnership as the co-host of this webinar. For many of you, EMBO, with its membership of more than 2,100 preeminent researchers, is undoubtedly a familiar and respected name in life science. EMBO is recognized for its dedications to promoting scientific excellence throughout Europe and beyond. And in this InnovaTalk webinar, let me introduce Professor Fiona Watt, Director of EMBO, who will serve as the chair of today's webinar. Professor Watt is also a member of the VinFuture Prize Pre-Screening Committee. We are also honored to welcome 2 leading figures in the field of regenerative medicines as our distinguished speakers. Professor James Bainbridge, Consultant Retinal Surgeon at Moorfields Eyes Hospital, London, and Professor of Retinal Studies at University College London. Dr. Cristiana Pires, Chief Executive Officer and Scientific Co-founder of Asgard Therapeutics. And for the sessions, we are also pleased to welcome a special guest, Dr. Linh Nguyen, founder and CEO of SmileScape, lecturer at Eastman Dental Institute, University College London, recently recognized as one of the UK Tech Women's 100 awardees. And now, to set the scene for our conversations, let's watch a brief trailer that provides an overview of what we will be discussing today.
Focusing on repairing, regenerating, or replacing damaged tissues and organs caused by disease, injury, and aging. Regenerative medicine is expected to become one of the pillars of future healthcare. Instead of merely treating symptoms, regenerative medicine aims to address the root cause of diseases, offering long-term recovery opportunities for patients. This field integrates various cutting-edge technologies, including cell therapy, tissue engineering, gene therapy, bioprinting, and biomaterials, driving significant advancements in modern medicine, rapidly advancing and is expected to become one of the pillars of future healthcare. To explore the many potentials of this transformative field, the VinFuture Foundation joins hands with the European Molecular Biology Organization to host the InnovaTalk webinar themed Regenerative Medicine. Sharing this InnovaTalk webinar is Professor Fiona Watt, member of the VinFuture Prize Pre-Screening Committee. Professor Watt is the leading scientist in stem cell biology. She established her first research group at the Kennedy Institute for Rheumatology in London, and then spent 20 years at the Cancer Research UK London Research Institute, where she helped to establish the CRUK Cambridge Research Institute and the Wellcome Trust Center for Stem Cell Research. In 2022, she became the director of the European Molecular Biology Organization and a group leader at European Molecular Biology Laboratory Heidelberg. Joining her on the panel are 2 distinguished speakers. Professor James Bainbridge is Consultant Retinal Surgeon at Moorfields Eye Hospital, London, and Professor of Retinal Studies at University College London. As a surgeon-scientist, his aim is to protect and restore sight by developing new treatments for diseases of the retina. His program of translational research extends from the laboratory investigation of mechanisms of disease to clinical trials of new medical and surgical interventions. Another distinguished speaker is Dr. Cristiana Pires, Chief Executive Officer and Scientific Co-founder of Asgard Therapeutics, a preclinical stage company developing in vivo direct reprogramming approaches for cancer immunotherapy. As CEO, Dr. Pires spearheads Asgard's mission to pioneer off-the-shelf therapies capable of recreating functional immune cells within the patient's body, overcoming the manufacturing and logistic hurdles of traditional cell therapies. And for this session, we are also pleased to welcome a special guest, Dr. Linh Nguyen, founder and CEO of SmileScaff, lecturer at Eastman Dental Institute, University College London. Her groundbreaking research includes the development of a patented technology involving scalable polycaprolactone macrobeads for stem cell harvesting, which has been successfully licensed, highlighting the commercial impact of her work. In 2024, she was honored as a Tech Women 100 winner in the UK, celebrating her contributions to technology and innovation. Beyond medical applications, regenerative medicine holds great significance for science and society. Progress in this field also promotes the development of biotechnology, pharmaceuticals, and biomedical engineering, helping to extend lifespan, improve quality of life, and reduce the burden on healthcare systems. Uncover groundbreaking advancements in biotechnology and regenerative medicine from leading experts in the field at the InnovaTalk webinar, Regenerative Medicine.
Let's welcome our esteemed chair for today's discussions, Professor Fiona Watt. Professor Fiona Watt is the director of the European Molecular Biology Organizations in Germany and a member of the VinFuture Prize pre-screening committee. Please join me in giving her a warm virtual welcome.
Thank you very much. It's a real pleasure to partner with the VinFuture Foundation for this webinar. As you heard, I'm the director of EMBO, which was founded back in 1964. And our mission is to promote excellence in the life sciences by creating an environment in which everyone can achieve their best work. You should take a look at the embo.org website, and there you'll see some of the programs that we're involved with. We're a leading academy where each year we elect members and EMBO associate members who are internationally expert. We fund scientists, particularly to promote mobility around the world. We also publish journals and innovate in open science. It's always interesting to get a perception of how science is conducted around the world, and to realize that no matter where we work, we all are driven by curiosity and a desire to explore life science. So when I was invited to put together the program today, it was easy for me to choose the topic of regenerative medicine. I've worked in the field of stem cells for many years, and what I find most exciting about the field is that really almost from the beginning, people have been asking the question, is this useful? This might be beautiful science, but could it be used to repair or replace damaged tissue? And it's a bit like back in the 1970s, the US launched a war on cancer, and people would say, well, cancer isn't cured yet, so that war failed. But that's actually a misunderstanding of the way science works. In the case of cancer, over the last 50 years, there's been steady progress through multiple approaches to reduce the incidence of cancer, diagnose it earlier, and improve treatments. And I believe that the field of regenerative medicine is very similar. We are working forward on many, many different angles. And as I hope you'll hear today, it's really very exciting and inspiring to see the way the field is developing. So today you will hear about potential and proven treatments which span cell and gene therapy materials, and also the remarkable underpinning science. So I've been looking forward to this webinar for some time now. I hope you will enjoy it as well. And I would like to end really by thanking James, Cristiana and Lynn for taking time out of their busy schedules to share their exciting work with you. So thank you very much.
We will turn to our first speakers and also our special guest, Dr. Linh Nguyen. She's currently a lecturer in biomaterials and allied subjects in Eastman Dental Institute, University College London, and founder and CEO of SmileScaff, a company dedicated to pioneering scaffold technologies for dental tissue healing. With her expertise in biomaterials, Dr. Nguyen brings a wealth of knowledge to today's discussions. Please join me in welcome her to the stage.
So thank you very much for the introduction from Professor Fiona and for the invitation. So I'm very honored today to talk about my current research interest in regenerative medicine through biomaterial and the cell therapies. For the collaboration purpose, please allow me to give a few slides for the introduction about. So UCL have 11 faculties. So we are under the faculties of medical sciences and the division of biomaterial and tissue engineering located at the Royal Free campus in London. So we have the well-established, the lab well-established on the equipment. We doing research supervising undergrad student and PhD student, including international and UK home student. We're doing teaching and we very keen on the global collaboration for increased networking. So the facility we have from the material fabrication to material characterization. We have the cell culture lab, and also at Royal Free campus, there is the animal study lab in there as well. So we have everything so the student can get very quickly, can get the data for the publication or for the research for collaboration. So I have been done several research, but today I would like to introduce to you three of the big research I'm doing now. So I am very interested on bridging the gap between the bench to the clinical application. So the first one is about the socket bone grafting. So after a tooth extracted, it leads to the significantly bone and tissue loss. So making the dental implant is challenging and uncomfortable for the patient. So I've been developing a material that can fill into the pocket. And then after that, the patient can do the dental implant. Then the healing process is very quick and it can help the patient bring the benefit to the patient and the dentist. So for this work, I found a SmileScaff, it's less than 1 year company. And because the market out there and we want to make the product made in UK, so that's why my SmileScaff was established last year. And because this work in the IP protection, so that's why I couldn't share that much about this technology. So the next one I would like to introduce to you that about a smart material for stem cells expansion and harvesting. And why do we need to do this? This is the manufacturing process for cell-based regenerative medicine. And the two steps is very challenging. It's the cell expansion and harvesting because it's a time consuming and it's very expensive, especially for cell harvesting. It's not yet the optimum quality of the cells. For cell expansion, traditionally, we use the D-flat or even though the multi-layer of the vessel. However, by using this method, we couldn't obtain the requirement, the large amount of the cells. So bioreactor is showing very potential for scale up the cells. So what we do, we culture the cell on the carrier. It can be microcarrier or macrocarrier, and then we put inside the bioreactor. So by this way, we can obtain the massive amount of cells for use for clinical application. So for cell harvesting, traditionally and a common use, we use trypsin. However, the trypsin is somehow it can degrade the cell membrane, it changes the cell phenotype, and there's so many disadvantages. So we found that a thermoresponsive polymer, when the cell detached from the surface of the thermoresponsive polymer, it still remains the cell quality. So that's why we came up with a project we are rafting the thermoresponsive polymer, it's called polynic balm, on the surface of the PCl polycaprolactone. And then we found that at 37 degrees, the cell attached on the surface of the system. But then when we reduce the temperature, the cell will detach happily from the surface without using any trypsin. So this work has been published and patented and licensed by a company for commercialization. So basically, we did the chemical reaction for the surface modification on the surface, and we test for the cell before testing for cell detachment. We need to do for cell attachment and proliferation. So we used human dermal fibroblast cells and mesenchymal stem cells, and then we put inside the bioreactor. So I just want to show some very highlight research in here, and we compare with trypsin as well. So by using trypsin, we can obtain more cells than by using the thermoresponsive polymer. However, by using trypsin, the cell viability is very low. It's lower than by using the thermoresponsive polymer. And also we collect the cell, and when we culture up to 1 week to see the cell recovery, so the cell obtained by reduced temperature, it shows that the cell growing faster than trypsin. So this is the result of the MSC. So in this case, we use the green fluorescent protein to clone inside the MSC cells in green color. So after reduce the temperature, the cell detached from the carrier and still remain the green fluorescent. So it means the cell is still healthy. And also we did the protein expression for the fibroblast cells and compare with the trypsin, and we can see that with the fibrinogen and laminin, the cell detached from reduced temperature, it still remain the protein expression. However, if we can see the picture on the trypsin, it's very weak signal that we can see. So it means somehow the protein is degraded by the trypsinization. And also it's very agreed with the Western blot, another method to see the protein expression. And just very interestingly, the collagen 1 in the cell membrane was not affected by the trypsinization. So, and then we also design the bioreactor, and I work with an engineer to build this bioreactor, and it's just only for macro carrier, my macro carrier use, and it shows that have potential for cell expansion. And also, in order to commercialization, we have to show that our material have ability to scale up. So that's why we investigate about like how to scale up the material. So by using this mixer, we can make like 1 kilogram of PCl conjugate with the thermoresponsive polymer. It's within only 24 hours, and it's all have been published for this work. And another research I'm very interesting is thermoresponsive polymer, but used for cancer treatment. So the polynic balm is not only have potential for cell harvesting without trypsin, it also have potential for controlled release of the drug. So my student Emily, she used the thermoresponsive polymer polynic balm to graft on the surface of the egg-shell membrane, and she lost the silver nanoparticle in the polynic balm, and it showed that at 37 degrees, the silver nanoparticle encapsulate inside the polynic balm, and when we reduce the temperature to 4 degrees, so the silver nanoparticle is released. So by that way, we can control the release. So on the right side, you can see that at 4 degrees, after 5 hours, the amount of the drug released faster and more than if we don't apply thermoresponsive polymer. So we can use this technique for any emergency. For example, if we need to release the drug, it's very quickly at the beginning. And also another student, Daoud, he drafted the polynic balm on the surface of electrospun PCl membrane and loaded with the nanocurcumin. And it showed that if you can see the blue line is at 4 degrees with the thermoresponsive polymer, after 2 hours, we can see most of the curcumin released from the system. So it can release very quickly. But then if we don't apply to reduce the temperature, there's no drug to release, so we can really control release of the material of the drug. And another project is very, very exciting. My excited project is have been working with the two PhD students, Antonia and Nuno. So they try to wrap the thermoresponsive polymer on the electrospun membrane. But this thermoresponsive polymer is work when we increase the temperature. It's not decrease the temperature. So we're grafting on the surface of the electrospun membrane, and we try to load the magnetic iron oxide nanomaterial. So our hypothesis is that when we increase the temperature to 43 degrees, the iron oxide will release because of the thermoresponsive polymer will swell, and it releases the iron oxide to the environment. And under the magnetic field is the magnetic iron oxide. It will heat up, and when it heat up, it will kill the cancer cells. So it's the hyperthermia treatment. So by this way, we try to develop the project for melanoma. Is a topical, and then if successful, we will try to it will work with another cancer disease as well, and it's an ongoing project. So for me, I think my overview of Vietnam, the current advancement in regenerative medicine, we have been seeing the success of several clinical trials on stem cells therapies, research and therapies, and especially there are so many institutes have shown that very successful for this. And also I have been observed there is an increasing of the publication in RegenMed in Vietnam in the last 5 years. Especially Vietnamese scientists show that they know the resource of Vietnam is using the natural material. So for example, there are so many publications and focus on the chitosan from some seafood, and the silkworm is very, very abundant resource in Vietnam. We should use that, and it's very successful in the last 5 years. And for me, I think we have to focus more in the smart biomaterial, especially like thermoresponsive or pH responsive cell assembly, cell healing, for example, because when the material that is smart material, then we can control the release. So everything, the dose and for example, cell release as well, everything, if we can control, then we can make the breakthrough. And also the bio 3D printing have been development, but the bio ink development, we need to focus more about that. And especially there are some publication show that they are working on the bio ink development, but we have to show how to scale up, how to make the large amount of the bio ink. That is also very important. And the sustainability in healthcare is very the government all over the world. There are so many countries they focus on this. So Vietnam, we have to speed up, and we have, if we don't do, we have to start to think about this and do some action about this for the Vietnam net zero target in 2050. For example, there's something I think we can work on this. For example, we have the sea, so we can use the seafood waste and turn it into the biomaterial. For example, the crab shell we can extract chitosan, or the cuttlefish we can extract to make the hydroxyapatite. And also the spheroid and organoid research is also, I think we need to consider, because in this case we can use the human cells for cancer research. Then we can limit the use of the animal study. And also this is culture meat and lab raw meat is happening right now all over the world. Actually, this is kind of regenerative medicine. So people growing cells in the lab and make the cell is binding together in the bioreactor, and then they turn into meat. So it's actually the other side of regenerative medicine, but then we can reduce the animal using. And AI, everybody now using AI, and I think it's also a very good tool that we can predict, and then we can reduce the number and reduce the time. We can save time, save money, and we can, it's very quickly we can get into the final research. And the last one I would like to share is about the establishment of the startup investment. Because why startup and spin out? Because the startup and spin out is commonly it bring the new technology to the field, and it can attract the investment and create a job. because it's more startup company, so more biologists and more students will study in this field. And I know that there's academic, they know that they need to do the translational research, but then they don't know how to do the step to do. So that's why the government or the private sector, we need to build something. We need to support the academic in the university, give more grant, and also the collaboration between industrial and academic, build some hub that, for example, the industrial then can go to the lab to share the lab in the university, and academic also can learn the step to commercialization. So that's one we can attract and then make the impact to the social, to the public. So the impact is very important. So hope I finish on time. So thank you very much for your attention, and I'm happy to receive any question later. Thank you.
Thank you, Dr. Linh Nguyen, for your wonderful insights. And next, we are pleased to welcome Professor James Bainbridge, consultant retinal surgeon at Moorfields Eyes Hospital, London, and Professor of Retinal Studies at University College London. Professor Bainbridge.
Great pleasure to be asked to speak at this event today. I've been very fortunate as a surgeon to be involved in the development of gene therapy, particularly translation of gene therapy from the early developments in the laboratory through to application in patients with inherited retinal diseases. And I thought today it would be most instructive just to provide something of a high-level overview of the progress in gene therapy for genetic eye disease, and to point out some of the successes and also some of the challenges that are needing to be addressed to take it further. So genetic eye disease of the retina is very important because it's common collectively. Inherited disease of the retina is common in children, and of course causes lifelong impairment of the sight in those individuals. But in addition, it's highly relevant to the older population, who not only are at risk of developing similar conditions, but also at risk of developing acquired retinal degeneration, such as macular degeneration, for which this technology may also be helpful, as I will go on to explain. But today what I wanted to just outline is some of the approaches to gene therapy and some of the challenges. So inherited gene defects may be addressed by a number of approaches, in particular gene supplementation, which of course is relevant for loss of function defects in recessive disease. And that's what I'm going to concentrate on in this talk. But I also want to touch on some of the opportunities and challenges with respect to treating dominantly inherited disease, which is typically associated with gene defects resulting in abnormal or toxic gain of function, oligonucleotides, and also more recently gene editing, which provides a cure. So of course gene supplementation and gene silencing are in many ways compensation for the genetic defect, whereas gene editing offers the possibility of not just compensating, but actually fixing the problem at the very highest level. So genes can be provided to the eye by injection of vector suspensions. And on the right here, showing how you can inject a vector suspension into the retina at the back of the eye here. And it's really done in this way in order to target the key cells, the photoreceptor cells and the supportive pigment cells that are directly implicated in the most common inherited retinal diseases. It is possible to inject naked DNA here, and there is some take-up of that, but it's very inefficient, and so the efficiency is improved massively by packaging first in a virus. And we typically use adeno-associated virus for this because it's relatively safe, relatively non-pathogenic, and can accommodate most of the genes that we want to include. Having said that, its capacity is a bit limited, and so it doesn't accommodate all of the genes that we'd like to be able to provide to the retina. So the eye is in many ways highly suited to gene therapy, because you only need a little bit of vector to transduce quite a large proportion of the target cells. You can study the structure the retina in detail in the living eye, and here you can see a scan, a cross-section through the living eye, almost at a histological basis, comparable to histology. And with adaptive optics techniques, you can see individual cells in the living eye because of its transparency. And this is very, very helpful when we're looking at targeting vectors to the eye, and indeed looking at the effects of those vectors on the outcome in terms of retinal architecture. But you can not just look at architecture, you can also look at function. So on the right here, I've just shown an image of a representation of the visual sensitivity in this area of the retina. So you can measure the sensitivity in terms of, or in fact, you can map the sensitivity in terms of function. So it's accessible to intervention, and it's accessible to study in terms of natural history and the effects of treatment in a way that is quite unique. And indeed, this has led to some notable successes in terms of gene therapy, and one is with targeting of a blinding condition owing to the deficiency of an enzyme called RP65, and this results in a loss of rod photoreceptor function. So in particular, young children are born with very poor night vision, and they go on to develop cone degeneration, which results in ultimately in very poor vision in both light and dark. But when treated early on, it can improve function substantially. So these are some of the results of the definitive RCT, showing that in the treated population in green here, there's a big improvement in the ability to navigate at night. And the population that was given deferred treatment, the improvement is deferred. So this is strong evidence of improvement in night vision in terms of its impact on the ability to navigate in dim light. This is reflected in a different test, which is called full field sensitivity testing. And this is a test which is easier to do. It involves basically flashing a bright light and asking patients to indicate whether they can see it or not. And here you can see with this relatively simple test, again, the population, the arm of the study that were treated initially improved substantially straight away, and the arm that got deferred treatment improved after that deferred treatment. So this is very powerful. In real life, this treatment in the sort of later childhood results in no obvious change in visual acuity, or indeed in visual fields, but it does result in an improvement in this FST, this visual field sensitivity measurement, and this is associated with anecdotal improvements in the ability to navigate at night. So this is consistent with a useful treatment effect, which is sustained after a single injection for many years. We've gone on to look at more recently to look at a similar condition, which is even more severe, affects children in early infancy and causes not just night blindness, but day blindness as well. And this is a condition in which defects in the gene encoding a protein called AIPL1 results in very poor function of photoreceptors. Here you can see a video of the injection underneath the retina. You may be able to see the bleb, like a blister here, of the vector making its way across the back of the eye to include the macula, the central part of the eye where most of the cones are affected. And this invasive technique is necessary at the moment to be able to target those photoreceptor cells in the central retina efficiently enough. And we found that in 4 children who we treated at the age of 1 or 2 years and followed up for 3 or 4 years, the sight of their treated eye improved really remarkably, from essentially undetectable to be able to read letters on a chart, whereas the untreated eye did not improve, or indeed deteriorated to become unmeasurable or having no light perception at all. So this has been very dramatic, recently published, and some other children, about 7 or 8 other children, have also been treated since then with similarly remarkable results. So in this particular scenario, there's a really dramatic improvement in sight, and the challenge now is to extend that to other conditions. This is an example of the impact in one of those children, impact on their ability to perform real life tasks. So this girl been asked to transfer crayons from one pot to the other. At the moment, she's using the eye which has been treated and the untreated eye which is covered, and you can see that she seems to be able to be guiding her movements with her sight really quite effectively. And she's subsequently asked to do the same task with her treated eye covered and using only her untreated eye. And here she seems to be struggling a little bit more. She's kind of realizing there that she couldn't see where the crayons were and had to resort to using her treated eye by peeping outside the mask. So this has been very successful in this particular conditions, and the aim now is to extend that to other inherited diseases associated with loss of function defects. So there's a range of recessive diseases, some of which I've listed here, which are currently the target in a number of different clinical trials. But since gene therapy can potentially establish expression of the therapeutic proteins in the eye after a single administration of the vector, this is also being used as a potential alternative delivery system, sustained delivery system for conditions which are not single gene defects but are often treated by repeated injections of a protein. And so in macular degeneration, for example, in repeated injections of VEGF inhibitors are used in many patients every month for many years, but the possibility that gene therapy can establish sustained expression in the long term is very attractive. Excuse me. So in summary, for gene supplementation therapy, this technique can achieve definite improvements in loss of function gene defects. It's expanding field of early phase and later phase clinical trials, and the first gene therapy, that is for RP65, is not science fiction, but actually is a licensed treatment and is a reality. Some of the challenges are that the dosing can be difficult to manage. The scope is limited by the capacity of the available vectors, AAV in particular, and it cannot address toxic gene defects. So to address toxic gene defects, the alternatives are gene silencing and gene editing. And see, these are some of the targets which are being addressed for gene silencing: Stargardt disease, RP, and other forms of Leber congenital amaurosis. And indeed, there is some evidence that repeat injections of antisense oligonucleotides can address a single substitution which otherwise causes harm in this condition. But because antisense oligonucleotides are short-lived, repeated injections are needed. So the alternative is to use gene editing in order to not just compensate for it, but to correct it at its root. And many people will be familiar with the new technology of CRISPR-Cas, in which a guide RNA is used to target the length of the mutation in DNA itself, and this is attacked by the Cas9 enzyme, and then repair mechanisms can lead to more or less specific deletions or substitutions. And indeed, in this condition I mentioned before, where there's a single substitution that leads to a loss of function defect, it's possible to delete that substitution using this mechanism in the living eye of human patients, with some evidence now in clinical trials of some definite impact. In fact, that impact appears to be reliably measured using certain sensitive tests, but it's been disappointing that the efficiency has been insufficient to actually result in life-changing benefits. So some of the challenges for gene therapy relate to the dosing and the efficiency, as I've mentioned, but also to the capacity and the immunogenicity of the vector, and the scalability and safety of that invasive surgery that I mentioned. And so with respect to the vector itself, there is some evidence that it can do harm. Here you can see some changes in the fundus. It's these little patches of atrophy in the retina which appear to be induced by the treatment. And this is a concern. It's not yet had an obvious impact on function, but does highlight the possibility that immune responses to the vector itself may cause harm or may limit the benefit of the gene therapy. Although we are still dependent very much on surgery to get the vector to the right place here, there are alternative, slightly less invasive ways of getting the vector to the back of the eye without having to go through the front of the eye, as illustrated in these diagrams here. It's possible to inject vectors into the vitreous, which is relatively accessible, but that's still relatively inefficient and appears to be pro-inflammatory. And the other issue that we need to address with respect to clinical trials of degenerations in general is that we need reliable outcome measures that can be measured in a timely way, and that's quite difficult with respect to degenerative conditions which are relatively slowly progressive. And we also need value comparators, so whether that's using the fellow eye, the contralateral eyes of control, or cohorts which are untreated, it leads to scientific and ethical questions, ethical considerations. So in summary, gene supplementation therapy is actually a reality. This is available worldwide for specific conditions. Gene silencing can improve sight temporarily, and gene editing offers the possibility of a possible cure. But I've outlined all the challenges as well. This is only effective for targeting of surviving cells. In advanced degeneration, we need alternatives such as cell therapies. We need to be able to control expression during the therapeutic window, protect against those immune responses I mentioned, and optimize trial design. So I've been very lucky to be involved in this, but it's actually the work of many, many teams and individuals, some of whom I've acknowledged here, but there are many more. Thank you very much.
Thank you, Professor Bainbridge, for your enlightening presentations. And last but not least, we welcome Dr. Cristiana Pires, Chief Executive Officer and Scientific Co-founder of Asgard Therapeutics. Her company is pioneering in vivo direct reprogramming approaches for cancer immunotherapies. Dr. Pires, we look forward to your insights.
Thank you so much for the intro, and also thank you so much for the invite to be here today. I'm very happy to present to you the work that we are doing at Asgard Therapeutics, and it's a bit different flavor, so it is still a gene therapy approach, but it is for cancer immunotherapy applications. And the concept is to bring this in vivo direct reprogramming of tumor cells and use it to restore immunogenicity. And so when we say reprogramming, we are actually really taking the learnings from the regenerative medicine and the stem cell fields on really engineering cell fate with direct cell fate conversion approaches. But we are using it really with the aim to recreate the therapeutic immune cells we desire directly inside the patient's body. And the idea with this is really to have innovative off the shelf, but there are still personalized immunotherapies. And I'll show you today some of the work around our lead program, which we called AT108, which we published recently, preclinical proof of concept. It's a very much tumor agnostic platform, so it actually works across the different solid tumor applications. But there's also opening up doors for additional pipeline programs in the same direct reprogramming setting. And the focus of the team today, and we are around 14 employees at the company at the moment, is really to push the lead program to start clinical trials by the end of 2026. And so our whole goal really started on trying to use these approaches to bypass the challenges with the traditional cell therapy approaches. And of course, those have been very promising, both autologous, and there's also some progress on the allogeneic cells, but they still come with a lot of complexity from the logistic and the manufacturing, and of course, the cost side of those. There's also an advent of, a promising new avenue, which is this in vivo cell engineering. And for example, chimeric antigen receptors are already being tested in the clinical trials to be delivery to the T cells directly in vivo. But the aim there is still just to functionalize or activate the cells that are already present within the patient. And we want to take this even a step forward. So we really want to completely create a cell that was not there from the get-go, so that we can really harness all the unique functional features of the given cell fate. And to start this approach, we really focused on how could we use this to bypass some of the really underlying challenges on cancer immunotherapy field. And most of you might be familiar that, of course, immune checkpoint blockade have been quite successful in changing cancer treatment, but still has been shown that the release of the T cell activity will only work if there's a proper antigen presentation and priming from the beginning. And tumors actually devise these strategies to both evade antigen-presenting cells from the tumor or to downregulate their own antigen presentation as a way to become invisible to the T cell activation. And so to bypass this, we were really inspired by the cells in our body, which are the best cells to do an antigen presentation process. And this is a really defined subset of dendritic cells, and these are called the conventional type 1 dendritic cells, and they are very efficient on showing the antigens on MHC class 1, but also MHC class 2, so both cross-presentation and antigen presentation. And they also secrete a lot of the chemokines and cytokines that together are very required and necessary to really recruit, but also activate and prime the cytotoxic T cell responses. These cells in our body, it's important to note that they are very short-lived and they are also very rare. So in the tumors of patients, usually less than 0.1% of the cells that are there are these CDC1 subset of cells. But yet this reduced number has been shown to be really critical and correlated with better survival for the patients, but also very important to mount an efficient anti-tumor immunity and response to checkpoint blockade, for example. And so our lead program really aims to do this. And how does it work? It is, in fact, a gene therapy. So it is a replication-deficient adenovirus vector that encodes reprogramming factors. And we are injecting it intratumorally to start off to really deliver these reprogramming factors into the tumor. And what happens is that then the factors can really convert the tumor cells, but also other cells in the tumor microenvironment to really now become and be converted into a dendritic cell, into a conventional type 1 dendritic cell. And by this, we force the tumor to now present their own tumor neoantigens on all the right professional machinery and really activate an immune response against itself. And this mechanism happens both locally, so there's a lot of changes in the tumor microenvironment with recruitment of these effector immune cells, but there's also a mounting of a systemic immune response that actually leads to an abscopal effect. So we see control of the tumor growth in the treated tumors, but also on the non-treated tumors. And so with this approach, we are really developing a product that is designed to be completely off the shelf. So it's always the same vector and the same factors across patients and indications. But the response is actually personalized because it's dependent on the antigens that each tumor shows within its patient. And so how do we get here? So it's actually a work that started since 2016, more or less, with a very fundamental biological question on what are the transcription factors that can really convert a non-related cell type into an antigen-presenting dendritic cell. And this was a classical screening process of different transcription factors, and we identified P1, RF8, and BAT3, which in short we call PIB, are able to actually convert mouse cells in the beginning, but then this was extended to human cells into this dendritic cell fate. And it was quite remarkable when we actually showed that this would work if we start from different cancer types and the cells become dendritic cell. And mechanistically, we have even shown that the 3 transcription factors, once overexpressed in the cancer cells, they actually really bind to chromatin of the target cell and induce a common dendritic cell gene signature. So there's a lot of genes that get upregulated. And then this leads to this cascade of events with upregulation of MHC class 1 and class 2, with co-stimulatory molecules being expressed, but also the typical cytokines and chemokines that dendritic cells also secrete. And so in the end, there's all this signature that is really critical to activate the T cell responses. But of course, then the question was, we can do this in the Petri dish. Can we really move this into an in vivo reprogramming approach that occurs within the living organism? And the first question was really about reprogramming, right? In the tumor context, which is usually very immunosuppressive, will reprogramming occur to the same extent? And then the other questions were more, how can we actually deliver those factors into the tumors in the patients, right? And will we drive a response in the end? And so to start with the first question, we really wanted to do it independently of the delivery to the tumor cells. And so we devised a strategy whereby we transduce the cells in the Petri dish still with a PIV vector or an empty control vector. And then after a short incubation, we take that cell mixed with different tumor cells in different ratio and put it into the living organism. And we have shown if we do this, for example, with human cells in a CDX model, and we take the cells at different time points, the cells were indeed reprogramming and acquiring the new phenotype in vivo. But if we use fully immunocompetent animals, we can actually look, is this driving some anti-tumor immunity? And it was very remarkable and exciting for us to see that indeed it does. And here I'm showing 3 different mouse models, all of them resistant to checkpoint treatment, and they have different profiles of how the tumors are infiltrated. But you can see that if we have in vivo reprogramming in the condition here just with a PIB vector, we have control of the tumor growth in some tumor models. Others, we even have complete tumor regressions already in a single agent setting. And as expected, is actually synergistic with checkpoint blockade. For example, when we combine it in some of the models, we get an improved efficacy in the models. And as I mentioned, we wanted to see, is this happening, something local happening here? And if we take out the tumors in these treated animals with a PIB on the right side, you can actually see there's a very dramatic change on the profile of infiltrates into the tumor with very dense clusters of lymphocytes being CD8, CD4, and even B cells, which actually even resemble tertiary lymphoid structures, which are also being described in the field to be important for an efficient anti-tumor immunity. But of course, then the other question was how to actually deliver these transcription factors into the tumors. And here we're very systematic, taking the learnings from the gene therapy field for regenerative medicine purposes. And we compare the original vector, which was a lentivirus in vitro, with the adenovirus and also adeno-associated virus through a lot of different experimental criteria. But we also looked, of course, from the precedent, from the clinical and regulatory side. And it's quite clear when you look onto oncology trials, in vivo gene therapy, adenovirus are clearly the strong contender here, as lentivirus and AAVs have, of course, been used either ex vivo or AAVs for a lot of other settings that not necessarily the tumor, right? And experimentally, then we validated indeed that the adenovirus can actually reprogram. And here we are showing different patient samples from different indications, both in a 2D, but also in a 3D setting. The virus can transduce, and they also induce a cell fate conversion into a dendritic cell, here measured with just 2 surface markers, but we have extended this to a lot of different markers and even transcriptionally. But also if we do it in vivo and we inject directly these vectors into a CDX model, we see that there's transduction, and also the cells that receive the transgenes actually reprogram also to the same phenotype of a dendritic cell. But then again, we wanted to show, is there an efficacy that comes after this treatment? And here again is one model, which is actually a very resistant model to checkpoint. In this case, is even resistant to both checkpoint blockade, anti-PD-1 and anti-CTLA-4 together, which we are using as a background here in these animals. And we injected our vector with a PIV 4 times in the beginning. And you can see that if we inject a virus that is a non-coding virus, so just a stuffer, there's already some expected immunogenicity that comes from the adenovirus itself, but we don't have any complete responses. But when we inject our transcription factors, we have 50% of the animals that are complete regressing the tumors, and even they survive long term. And here we wanted to really see, is there a systemic response that is mounted? And indeed, if we re-challenge the animals first subcutaneously or then intravenously, the animals were actually protected against the metastatic outgrowth, which tells us that this initial kind of treatment window and burst of these induced dendritic cells is sufficient to actually drive this long-term immune response. And of course, at this point, we wanted to understand how many cells do we actually need to have an efficient response here. And so we went back to the setting where we actually have 100% of the animals regressing the tumors. And here we control the number of cells that we are putting there. And we could see that if we go down on the number of cells we have being reprogrammed in the beginning, we actually see a very clear dose response. So the more cells we have, the better response we have. But also we could recapitulate that less than 2% of these cells in the beginning are actually sufficient to mount this response, which kind of also correlates with what is known from the patients that a very reduced number is sufficient. And so this then led us to really select the lead program, the 80-108, which is now actually undergoing more advanced preclinical development. So we have been discussing with the regulatory agencies, and we are confirming the manufacturing process. And after that, of course, we need to do the formal safety and tox studies, aiming to start the first in-human studies towards the end of 2026, beginning of 2027. And there the aim is actually to test this in advanced solid tumor, so as a basket trial, first on a dose escalation to prove the safety and tolerability of the approach, but then expand rapidly to expansion arms in specific patient segments where we can really show for the first time that an in vivo reprogramming works. And here we are actually keeping arms in the combination setting because we have seen synergy, but we also looked at patient populations that could receive this in a monotherapy, so we can really show the advantage of our approach. And we have identified 2 of those settings, one being the recurrent metastatic head and neck squamous cell carcinoma. There's a fraction of the patients that usually does not respond to checkpoint, and there's very poor prognosis. And the other one being the metastatic microsatellite stable colorectal cancer, which also do not respond to checkpoints. And in both these settings, there's evidence that the CDC1s on antigen presentation play a critical role for the tumor evasion mechanism, and that's what we are trying to reverse. And with that, I just wanted to conclude that in reality, to push a first-in-class immunotherapy forward, this is clearly a team effort, and I just want to stop by acknowledging everyone that is involved, from our investors, grant awardees, but also the extended scientific and development team. Thank you very much.
Many thanks, Dr. Pires, for your thoughtful contributions. And now, to wrap up our discussions, I would like to invite our panelists back to the stage for a brief panel reflection. This will be a dedicated 5 minutes for them to share any concluding remarks or insights before we move on to the highly anticipated Q&A sessions. Please join me on the stage. The stage is yours now. Professor Watt, can you please start the panel discussions?
Yes, to start with a reflection on this webinar. First of all, I'd like to thank all of you for really inspiring talks. But in each case, it strikes me that the basic science, in a way, is such a small part of getting to something that is going to work in patients. And I think, for me, that has always been the challenge of the stem cell field. People will publish their high-profile paper, and they'll say this offers treatment for X, Y, or Z. But as you've shown, it's a very long, tough, and expensive journey to make something that really works. So am I right in that reflection? I think, James, you might be well placed to answer that, because after all, you're a surgeon, so you're literally seeing the patients on a regular basis, and you must find it just difficult when you see a child who is born with a particular genetic defect, and you know in principle how to prevent blindness, but the steps and the expense for treatment are hard.
Yeah, indeed. So, of course, in principle, the supplementation gene therapy for many of these conditions is relatively straightforward. But of course, in practice, the situation is very different. There are, as I outlined, a few unique scenarios in which particular gene defects are amenable to gene therapy, gene supplementation in that way. And it's possible to measure the benefit in a way that is timely and compelling. It's taken a lot of work to get to that stage, and I think it's going to take a lot more to be able to extend the potential benefits into other conditions. And that's for a number of reasons. It's related to generating treatments which are efficient enough, and also demonstrating the benefits, and again in a timely way. So there are only a few situations where one might expect to see an improvement in function and an improvement in sight. In the majority of instances, it's optimistic to expect that, and in reality, it's going to be a question of measuring protection of sight over a long term. And of course, that introduces another challenge, because that makes for trials which are going to be lengthy and expensive and difficult to justify. So I think those are some of the challenges that we're now facing in terms of gene therapy. I might add, with respect to cell therapy, there's good evidence of stem cell treatments being very effective on the surface of the eye in terms of the epithelium, and that is a mainstay of treatment for a lot of corneal eye disease. What remains the challenge, but potentially with massive benefits, is to achieve a similar thing with the nerve cells in the back of the eye, in terms of replacing and regenerating photoreceptor cells. And those photoreceptor cells are not surviving in isolation, but they are dependent on a number of supporting cells, which have to be taken into consideration when designing new treatments. And so there are lots of challenges to be addressed. But I think that, as I think you said earlier, Fiona, there has been gradual progress, and I think that's what we can expect.
And if I can just build on what you said, for some of the diseases that you're treating, the number of patients is really small. But I would contrast that with Cristiana's work, where potentially you have a very large number of cancer patients. And I noticed that Cristiana and Ling have both started companies. So my question to Cristiana is, was that the only way to push into the clinic in your case?
I can start while Lynn thinks about it, right? So in our case, right, Asgard was even started, we are 3 scientific co-founders, right? And this was in reality my postdoctoral project back in the day, right? And at the time for us, I think it is actually possible to push this forward just as an academic, right? There are, of course, examples even here in the room, right? Or people trying to push those forward. What we understood at the time is that our concept was so unique and differentiated from what everyone was doing, that we thought that to have proper funding, or as much funding as we could, to allow us to move fast, but also have sufficient funding to de-risk this before we move to the patients, right? In order to try different vectors, try different approaches as much as we could, that we actually need a substantial amount of money that we couldn't get from grants that are accessible for academics as such, right? So I think inevitably funding is a big kind of gatekeeper for a lot of the things, right? And we kind of thought that it might be, it might improve the chances of us being successful when we get to the patients to have a bit more funding, but also I think the access to the know-how and being able to attract, people from the industry that have done a lot of those things before was also a critical kind of motivation for us to try to get and start a company as such, right? Yeah, so I think it also comes from the personal motivation itself, that I saw myself more as an entrepreneur, and my co-fellow founders were also aligned with that kind of ambition of start a company to take the science to the patients, right? But that's our kind of leading force and motivation, is always to bring this to the patients. Yep.
Okay. And Lynn, what was your motivation in setting up a company last year?
Yeah. So actually, I'm still academic. And so I'm 50% academic and 50% entrepreneur. And I think when establish a company, it pushed me to work with the patient. So also we have some, like we doing BPI, like patient public involvement with small group of the patient. We design the magnetic iron oxide hypothermia treatment. And we ask the patient, like, for example, if you want to do, you want high dose, but the less time to go to the hospital, or you refer to have low dose, but then you travel to the hospital more. So the patient opinion actually will help us. So eventually we will design back into the research, we will design like to make the high concentration or low concentration. So it supports to each other. So I like doing research, and also I like to see my product can be out there, somehow can help people healing faster. So that's why it's bring together. I love, and also I like working with students to support them. So that's why I think it's not like more entrepreneur or more academic. It's both. It can support, but eventually it spend a lot of time actually, because like to different work we have to do, like funding, to different kind of funding we have to deal with. So yeah, that is the motivation to establish the company.
And my last question to the panel before we open it up relates to getting regulatory approval to go into patients, because the treatments that the Cristiana and James have worked up would benefit patients in all countries around the world. But to get those initial clinical trials, you have to go to individual regulators. So am I right that that is an issue? Is there a way that things could be improved in the future to make treatments accessible worldwide earlier?
I think, clearly as international collaboration is going to be important. And I think in terms of the EMA, for example, that had been very helpful until Brexit. There've always been issues, differences of opinion between GTAC, for example, and the FDA, and I'm afraid to say I'm not sure that's going to improve. So I think you're right that it's going to be a challenge, but clearly it would be very helpful to have some sort of consensus internationally on what's required, because I think it does make things it does introduce additional challenges if the goalposts are different in different territories. Absolutely.
Cristiana, would you like to add to that? I notice you're based in Sweden, but you're getting FDA approval.
Yeah, so it's actually, so we are aiming to have the opinion of the FDA at this early time point, right? But the clinical trials are planned to start first in Europe, but then, of course, be prepared to then move them to the US, right? From, it's important for us to try to be aligned as possible to avoid, as James is saying, that sometimes there's a bit of misalignment, right? So even though regulators are not necessarily aligning themselves, we as a company are trying to align what we are doing to comply with both regulations at the same time, right? So it creates an extra complexity on the academics or companies that are developing it, right? Yeah.
Thank you all panelists for wonderful final remarks, but however, we are a little bit short. So I would like to move on to the next part, and so that we could come quickly to the highly anticipated Q&A session so that you can share more with our audience. And now, while our panelists take a well-deserved 5 minutes break, we invite you to watch a short video tutorial on submitting nominations for the 5th season of the esteemed VinFuture Prize. Keep an eye out for the video and we'll come back shortly.
Greetings from the VinFuture Prize. Please find instructions for the nominator following these steps. To begin, you can access the nomination portal at online.vinfutureprize.org/nomination, or you can scan the QR code you see on the screen right now to open the nomination webpage. On this webpage, you can download the instructions and the endorsement letter template. In the instructions, you will find the complete nomination form, where you can draft the nomination whenever you have time. Then, you can copy and paste the draft into the online portal. Please click on Sign Up, and upon successful registration, you can simply use your ID and password to log in. After login, please click Nominate Now to start a new nomination form. Subsequently, you can see 7 content tabs on the nomination form. To begin the nomination process, on the first tab, please select the prize category as well as the type of nominee, whether it is an individual nominee or a group of nominees. Then click the Start button to proceed to the second tab. And we would like to call your attention to tab number 5 and tab number 7. On tab number 5, your evaluation, you're invited to give your evaluation of the nominee's invention, including its groundbreaking discovery, scientific fundamental knowledge, specific approach, uniqueness, and advancement. Then you are required to clarify if the invention can be transferred to a product and highlight its most significant socioeconomic impacts. This includes the number of beneficiaries, such as people, countries, and continents. Additionally, please explain how you believe this invention has or will potentially transform the everyday lives of millions, if not billions of people. Next, please provide information on the history of the invention, such as when and how it started, and who else, except the nominee or nominees, was involved in initiating or contributing to the invention. About tab number 7, supporting documentation and evidence. There are several important documents that the nomination portal and form require, and we would like to call your attention to 3 critical items. The first one is the CV of your nominee or the CVs of your group of nominees if you are nominating a group of people. The second important item is the endorsement letter. 3 endorsement letters or more are strongly encouraged, but 1 is mandatory. The third one is the additional form of nominees. Please be as specific as possible when providing the information. Additionally, you are strongly encouraged to include any valid intellectual properties, publications, patents, or other supporting documents that provide evidence for the nomination. Finally, after filling out and providing all the content in the 7 tabs, please click on the Submit button at the bottom of the page to submit your nomination. If you have any questions or you face any technical issues, please email us at secretariat@vinfutureprize.org. We will be more than happy to assist you in your nomination process. Thank you very much.
Before moving on to the most awaited Q&A sessions, we would also like to share an important announcement from outside. The VinFuture Foundation is now calling for nominations for the 2025 VinFuture Grand Prize and 3 special prizes. The nomination portal is now open until April 17, 2025. We also remind our nominators of the VinFuture Nominators Recognition Programs, which acknowledge their contributions in identifying transformative scientific achievement. And now is the moment you have all been waiting for, the Q&A sessions. It's your chance to delve deeper with our esteemed panelists. Please feel free to actively participate by using the raise hand functions. And for those who prefer a more indirect approach, we encourage you to use the chat box. And now I'm receiving many questions from our audience. And to start the Q&A sessions, I would like to ask Professor Fiona Watt, our chair of today's webinar, to answer my first questions. Professor Watt, how do you see the role of interdisciplinary collaborations, such as between stem cell biologies, biomaterials, and bioengineering, shaping the future of regenerative medicines?
Thank you for the question. It's obvious that an interdisciplinary approach is necessary to push treatments into the clinic, but it's something that is not so easy to achieve. In my view, sometimes it's very important that the 2 or 3 partners have aligned professional and personal goals, even if on paper it might seem like a great collaboration, it can fail at that very human level. I also think it needs to be realistic, and it needs in the early stage to attract funding. I think that it is much harder for 2 applicants in different fields to be funded on one project than either individually, because what happens is that when evaluating the grant application, there will be criticisms from both sides rather than an appreciation that the partners are trying to do something different. So it's interdisciplinary our work is really important, but it's not as easy as writing it down as an aspiration.
Thank you, Professor Fiona Watt. And now I have received questions to Dr. Linh Nguyen from Dr. Trương Văn Ngọc from Moffitt Cancer Center, Florida. Dr. Linh Nguyen, the trypsins looks like almost kill all the stem cells that express the GFP. Did this happen for other cell lines? And can those detached GFP expressed stem cells be growth back?
Thank you for the questions. Actually, the trypsin didn't kill all the cells. Actually, it just somehow it slowed down the cell growth. It still helps the cell. That's why right now it's still we're using trypsin. So it didn't kill all the cell. I think somehow maybe some of the research maybe make the version is not so clear. And so trypsin is still work, but just not didn't the cell is somehow didn't remain the quality, the protein expression. And the second one is, of course, like after, for example, even though for the GFP MSC cells, after the cell detached from the thermoresponsive polymer or trypsin, we can collect the cells and re-roll, and it still remain, the GFP is still there. So I have done, and I saw that it's quite long time, it's quite stable, so it's possible.
Thank you, Dr. Linh Nguyen. Although there are many other questions, however, since the webinar is limited in time, all the article questions, please send to VinFuture Foundations through the email secretariat at vinfutureprize.org. And now, as we conclude today's webinar, we would like to extend a sincere gratitude to our esteemed panelists, Professor Fiona Watt, Professor James Bainbridge, Dr. Cristiana Pires, and Dr. Linh Nguyen. Your expertise and contributions have provided invaluable perspective and on the transformative potentials of regenerative medicines. We also want to thank our wonderful audience. Your engagement and thoughtful questions have undoubtedly enriched these discussions. And a reminder to our wonderful audience, the VinFuture Foundation is now calling for nominations for the 2025 award season. The nomination portal will be open until April 17, 2025. As part of our nominator recognition program, 5 outstanding nominators make significant contributions to the 2025 VinFuture selected to participate in Future SciTech Week and all expense covered by the foundations. As we wrapped up this InnovaTalk webinar on regenerative medicines, we appreciate your continued support and enthusiasm in this important conversations. We look forward to exploring more groundbreaking advancement with you in the future. Until then, best wishes to all, and we hope to see you at our next event.